Yesterday we were talking about a dihybrid cross, which is pretty much a fancy way of saying you have two heterozygotes, which means that there's different alleles. Dominant, recessive. And that's about it. Genes code for the same protein, but one's dominant and one's recessive. And you put them on the edges of the square. So in looking at it, I'm going to put dominant and recessive, right? Dominant and recessive. Yesterday we came up with this. We had a homozygous dominant, a homozygous recessive, and two heterozygotes. This is how you kind of figure this out. It's 50-50 because our parents both were heterozygotes and they both gave off a blue and a red. In this respect, this is a heterozygote or homozygote dominant, heterozygote, homozygous recessive. But the way the probabilities work out is that you can say it's 50% heterozygous, 25% homozygous dominant, 25% homozygous recessive. Okay, this is where it gets cool. Since dominant and recessive, since dominant overtakes and covers up recessive, you have 75% of them will look blue, meaning that their phenotype will be blue. This, all by itself, only 25% of them will have a phenotype expressed as red. And in looking at that, you have a 75% to 25%. That's actually how this cross, that's very, very popular, works out. 75% blue, 25% red, 25% homozygous dominant, 25% homozygous recessive, and 50% heterozygous. That's as simple as I can possibly put it for the basis of a dihybrid cross. Want to practice some of this by yourself? You're more than certainly welcome to. Try to figure out the probabilities of some other crosses. I'll cover a couple of those quickly tomorrow.